Plastic-eating fungi, nature's solution to plastic pollution. There are 11 classes of fungi discovered that could eat plastic. These fungi use special enzymes to break down plastic into smaller pieces, turning something harmful into something nature can handle. Moreover, certain fungi such as edible mushrooms as they eat plastics, they not only contribute to environmental sustainability, but also have the potential to generate food products, creating a dual benefit. The mushrooms produced could be used for human consumption, adding economic value to the process. How do fungi eat plastic? There are 4 steps. First one is attachment of fungi to polymer substrate. Second one is fungi colonization on plastic substrate. Third one is secretion of extracellular enzyme. And the last one is mineralization. Let's discuss about the steps in plastic metabolism by fungi. Attachment of fungi to plastic substrate. Fungi is made up of a network of tiny, thread-like structures called hyphae. Each hyphae is like a microscopic tube that grows outward, spreading through soil, wood, leaves, or even plastics. As the hyphae grow and connect, they form a large, dense network. This network is the mycelium. During attachment to the plastic substrate, it produces a protein called hydrophobin. Hydrophobins are small, globular, surface-active proteins produced by fungi. They are unique because they can assemble into tough, water-repellent coatings at surfaces such as air-water or solid-liquid interfaces. These proteins have the remarkable ability to change the surface properties of materials, making them either more water-repellent, hydrophobic, or more water-attracting, hydrophilic, depending on how they are arranged. Hydrophobins perform many important roles, helping hyphae grow into the air, allowing fungi to stick to surfaces like soil particles, plants, or plastics, forming protective layers around spores and fruiting bodies, assisting in biofilm formation. Structurally, hydrophobins are rich in cysteine amino acids, which form strong disulfide bonds, giving them high stability. Between 7,300 and 50 amino acids present in hydrophobin. Colonization. Colonization refers to the biofilm formation on plastic by fungi. The fungi cover the entire plastic substrate by extending and penetrating their hyphae into the material. Extracellular enzymes produced by fungi. Fungi produces 2 types of enzymes. First one is hydrolase, and the second one is oxidortictase. Hydrolase consume the plastic by hydrolysis reaction. There are different subclasses for hydrolase enzyme, such as esterases, which is used for hydrolysis of ester bond in polyesters. For example, cutinase and lipase are subclasses for esterases. Protease, which is used in hydrolysis of polyamides. Urethanase, which is used hydrolysis of urethane bond, or polyurethane. And last one is amidase, which is used for the hydrolysis of polyamides. The fungi responsible for secretion of hydrolase enzyme are given with degradable plastics. Lacase. Lacases belong to the enzyme class oxidortictase. It oxidizes phenolic and aromatic compounds by reducing O2 to H2O. Fungi secreting lacase enzyme with degradable plastics are given. Hydroperoxidase. Hydroperoxidase also belong to the enzyme class oxidortictase. It uses H2O2 to oxidize various substrate including plastics. Lignin peroxidase and manganese peroxidase are the main examples for hydroperoxidase. Fungi secreting hydroperoxidase enzyme with degradable plastics are given. Now let's see the plastic metabolism by fungi. Polyurethane has taken as a plastic substrate. Aspergillus flavus is a type of filamentous fungus, widely found in soil, plants, and nuts. It can produce an enzyme called urethanase, which is capable to hydrolysis polyurethane. Active site of the enzyme is represented by catalytic triad of amino acids, such as serine, histidine, and aspartate. The catalytic triad interact with the carbonyl group in polyurethane, form an enzyme substrate complex, which can undergo hydrolysis in presence of water, gives diol, carbamic acid, and regeneration of enzyme. The carbamic acid changes to diamine by the release of carbon dioxide by a spontaneous reaction. Mineralization is the final step of fungal plastic metabolism. The so-called plastic fragments absorbed by the fungi. It is called assimilation. Inside the fungal cell, the molecules enter a metabolic pathways like citric acid cycle, and the final products such as carbon dioxide, water, methane, biomass, etc., released into the atmosphere.